Black, Editor-in-Chief of ScienceBoard, and I'm thrilled to be here today with Miriam Merad, who is the Director of the Precision Immunology Institute at Mount Sinai.
So there was two sessions. There was a session yesterday where I talked, a session this morning that I chaired, and in both sessions the focus was myeloid targeting. So most of the immunotherapy most of the immunotherapy strategies are focusing on activating a group of immune cells called T cells, right? So how you can reactivate these T cell effector functions so that they kill tumor better. The focus of my session and my work is really another group of immune cells called myeloid cells. And the reason why we are interested in them is because they are the structure of T cell function. So it's really a strategy that is a little bit synergistic with all the activity, but the activity has been so far very, very focused on T cell. There's a lot of data that suggests that all these myeloid cells are also contributing quite significantly at dampening or regulating anti-tumor immune response. And this is what the focus of the two sessions were. Well, I think we still don't know all the immune system is a very dynamic system, right? So the same molecule can be stimulatory or regulatory because there is all this dynamic system where you have to reach a balance, an equilibrium, right? So you constantly have to activate, but while you are activating a response, you have to regulate a response. And so most of the molecules that we are focusing on probably have at some point two functions. One is activating and one is regulating. So it's going to be very, very important to capture as much as possible information about all the expression of this molecule during tumor progression. Also understand some of how some of these pathways are working together. There is I think it's possible to expect that targeting one molecule, let's say with the checkpoint, with the PD-1 checkpoint CTLA-4, will resolve, right, and will promote effective anti-tumor immunity in all tumor and all different states. So we still are in the process of accumulating information about all these different pathways, and the challenge is a highly dynamic system. Well, so COVID somehow, because it's still an immunological question, right? So it didn't hamper research as a whole, right? Because the research toward the vaccine can be repurposed, in fact, quite significantly to the cancer field. It did, however, slow down some of the projects that were not vaccine related, and it affected, for example, the clinical trial efforts quite significantly. But I have to say, I don't think that in the long run we will remember COVID as a big impediment, or I think these things have continued to progress quite, at least preclinically. Many of us have used, in fact, the fact that we are immunologists to continue some of our work on cancer, because a lot of this work was also useful for the COVID-19 effort. There is a big focus on vaccine, but there was also a focus on modulating inflammation in patients with COVID-19, and all these pathways that modulate inflammation are somehow also utilized in cancer. Different reasons. The opposite in cancer, you want to activate them. So a lot of this work can be repurposed. So I will not, in contrast to other fields, right, of science, I think cancer immunology is probably the field that was affected the least by the pandemic. What's exciting is to try to identify synergy between different pathways, targeted pathways. So I think we have to learn to use this combination treatment. The big transformation in treatment of cancer has been combination chemotherapy. That when we learned that we could use different chemotherapy agents, did transform, the outcome of patients with breast cancer, for example, and many other cancers. So we have to learn to do exactly the same in immunotherapy, and not only with with immune target, but potentially also combining with targeted therapy that will target tumor cells directly. Many of the targeted therapy are also potentially modulating immune cells. So we have to learn how these targeted therapy that are targeting specific mutation in tumor cells, right? Now I'm not talking about the immune compartment. Those pathways are also used by immune cells, and learning how we can potentially harness some of this targeted therapy to activate the immune system is really fascinating. So it's a fantastic, we are at a historical time, I think, because in addition to this revolution of immunology, there is a big revolution in technology that enables us, for the first time to really have a granular understanding of all these molecules that are being expressed or downregulated in tumor lesions. So it's up to us to design, the right type of study, and of course we have to raise money to be able to do this study. So the future is combination between different immunotherapy strategy, combination with targeted therapy. It's possible that chemo could still play a role, but really figuring out in a very rational manner how to combine is really the future. So the big focus of the lab now is to use to do experiment in patients, So really do these type of studies where we will start building knowledge of how the immune microenvironment and the tumor responds to immunotherapy. And we are really using a lot of technology, high-end and high-dimensional technology on patients. And the design is really this small cohort of patients, deep science, to understand response to immunotherapy strategy. So we've built a fantastic platform, and we are using what we call this window of opportunity trial. So this pre-surgical setting where patients can receive immunotherapy agent just prior to surgery. And then you will be sampling the lesions prior to the treatment and after the treatment by analyzing the surgical lesion. And that allows you, because you have a lot of tissue access, it really allows you to do a lot of type of measurement that are extremely informative. So we are really focusing on that platform and then this is used to generate hypothesis. As I said yesterday, you generate hypothesis and then we bring it back to the lab. And of course, my lab focuses on myeloid, but I also share this data with a whole group of collaborators at Mount Sinai where I work, and many of us will be looking in detail in animal models at different lineage, and then we come back to the patients, and then we go back to the animal. It's really this platform that we built in recent years as very technology-driven and immunotherapy-focused that we are very excited about. Very exciting time. Yeah, so I am passionate about it indeed because, first, it's a very expensive endeavor and it's a difficult endeavor. So not all of us have to redo it, in your own lab, and redo this type of profiling, extensive profiling that is so useful. So we could really, if we were smart, I think, somehow we could really put all this data together, because the profiling, of course, it's proprietary. You can feel very passionate about it, but really the work starts there. When you have all this data, what you need to do is the road is still very, very, very long. So if you can just save time and really use that knowledge, like an atlas, right? An atlas that everyone can use, and all of us are going to come up with different hypotheses and potentially different targets. But if we all, if we are, and I am generating atlases, and many of my colleagues are, but really, if we were to put the data together, we will be much faster in going and validating many of these hypotheses that I was just talking about. So I'm hoping many of my colleagues are doing it. Some of our colleagues are not doing it because their institution or their government doesn't allow them, or because they are industry, and industry sometimes is reluctant to let go these data. it's wrong because really the interpretation is very vast and you want people to look at this data. Initially, when we were profiling, with single-cell profiling of our human lesions, my institution also wanted to protect those type of data. Having that data set is just the beginning, right? You have, and we knew that should not be appropriate. What is proprietary is then the type of experiment, the focus, the prioritization of pathway of target that you are going to study. You have plenty of space to continue to develop. So we need to share so that we can move faster.
Yes, absolutely.
And patients are still sick.
I agree. Collaboration is the way forward.
Exactly.
Okay, awesome. Well, thank you, Miriam.
Yeah, my pleasure.